Start.
Sir.
Start, sir.
Okay, sir. Good morning, dear students. Once again, I welcome you all for this live online webinar class for the subject of zoology. This is Kumar, PGT in biology from TSRS&JC, Lingampally. Dear students, in the part of the revision of our zoology classes, we have been learning the important questions, 2 marks, 4 marks and 8 marks question from many days, isn't it? So today we are going to learn about a new chapter. Let's see what we are going to learn. Today we will be learning about immune system. Okay, immune system, it is the 4B chapter, okay, from your second year zoology. Let's get into the class now. So 2 marks question. First of all, we will see the 2 marks important questions, then we will go to the 4 marks. There is no 8 marks question that is expected from this chapter. Okay, only 2 marks and 4 marks questions are there. Let's see the first question. You just follow along with me. See the slide. It is there. Define the terms immunity and immune system. Okay, it is most prevailing term, isn't it? So due to this corona pandemic, everybody are discussing about immunity. Those who have strong immunity, they are getting sustained, they are getting prevented from corona, isn't it? So let's know this is what is this? Immunity means the ability of an individual to fight against the disease causing organisms. Okay? Ability of an individual to fight against the disease causing organisms is called immunity. దీన్ని తెలుగులో రోగ నిరోధక శక్తి అంటారు. Let's see what is the immune system now. Immunity is the ability, okay, and immune system means the network of organs, cells and proteins that protect the body from harmful infectious agents such as bacteria, viruses, animal parasites, fungi, etc. It is called immune system. Immune system is the group of organs, cells and tissues of our body which are involved in the immunity. Okay, so these organs are important because the chemical reactions, biochemical reactions take place in these cells and organs. Okay, let's go to the next question. Before going to the next question, let's see the difference between immunity and immune system once again. Okay, see what is immunity. Immunity is the ability of the body to defend against the disease causing organisms. Okay, so basically immunity means the defense mechanism, and immune system means it's a complex group of cells, tissues and organs. Together they help the body to fight infections and other diseases. What actually immune system does? Immune system identifies the self and non-self substances. Okay? These non-self substances are nothing but microorganisms, toxins, malignant cells, etc. Father of immunology is Edward Jenner. Okay, Edward Jenner. He is the father of vaccination as well. Immune system means it is our body's immune system. What it does, it identifies the self and non-self substances. Self substances are nothing but the substances which are created by our body. Non-self substances means the substances which are the foreign particles. They are from outside of the body. Let's see the next question. Second, see, write the names of any 4 mononuclear phagocytes. Examples of mononuclear phagocytes. These are located in the tissues mainly. According to the tissue it is located, it has got different names. So examples: Kupffer cells, microglia cells, osteoclast cells and synomial cells. Let's see where they are located now in the next slide. See here, mononuclear phagocytes. These are called MNP and they are also called as localized macrophages. Okay, these localized macrophages are mainly important to give protection in the tissues. Okay, if any bacteria or foreign particle goes into our tissues, these mononuclear phagocytes identifies them and engulf them and destroy it. Okay, this engulfing a foreign particle and destroying process is called as phagocytosis. Since they carry out phagocytosis, they are called phagocytes. Okay, phagocytes in normal terms we call it as cell eating. Okay, see here, bone marrow of macrophages, peritoneal macrophages which are present in the body cavity and peritoneal cavity. Bone marrow macrophages are osteoclast cells. Okay, osteoclast which are present in the bone, right? Next, alveolar and branchial macrophages are existing in the lungs. Langerhans cells, they are existing in the Langerhans cells existing in the skin, and placental cells, okay, they are existing in the placenta. Intraglomerular mesangial cells, these exist in the kidney. Fat cells are also there, they are present in the adipocytes, adipose tissue. And splenic red pulp macrophages protect the digestive system. Uterine macrophages are existing in the uterus. Connective tissue histocytes located in the tissues, body tissues, and blood monocytes existing in the blood. They are the blood phagocytes. Next, interstitial macrophages located in the intestine. Myocardial macrophages located in the heart, and Kupffer cell, just now we have studied this, it is located in the liver. Microglia, these are the phagocytes existing in the brain. So these are all different kinds of localized phagocytes. Okay, localized macrophages. These are also called as mononuclear phagocytes. Hope you understood about this. Let's go to the next question. Colostrum is very much essential for the newborn infants. Justify. Colostrum means what? First of all, colostrum means the early milk that is produced by the mother just after the delivery. Okay, it's called colostrum. Why they are most important? Because the colostrum secreted by the mother during the initial days of lactation. Lactation means the secretion of the milk. Okay, it has abundant immunoglobulin A antibodies, and they protect the infant. So why they are most important? Because they have antibodies. Okay, their immunoglobulin A antibodies are there. They help the infant to fight against the diseases. Okay, let's go to the next question, that is fourth. Differentiate between perforins and granzymes. Perforins and granzymes, these we study about these in the cell mediated immunity. Okay, so what are these perforins? Perforins form pores in the cell membrane. Okay, so what do? They make pores to the infected cells. Sorry, not infected cells, the bacteria or the foreign particles. What granzymes do? Granzymes enter the infected cells through these perforations and activate certain proteins that help in the destruction of infected cell. Okay. Perforins makes the holes to the infected cell, and granzymes goes inside of the cell. These are the substances. What do? They destruct the infected cell. Why the infected cell has to be destroyed from the body? Because infected cell, if it grows with all the microorganisms, other cells which are present beside also gets affected. So to protect from other tissues, infected tissues are completely destroyed by our own immune system. This is also called as apoptosis. Apoptosis means the death of infected cell. Okay, death of infected cell or killing of the infected cell is called as apoptosis. Hope you understood. Let's go to the next question. Explain the mechanism of vaccination or immunization. Vaccination and immunization. Let's see what is this mechanism, why we have to take vaccines. It is also the prevailing term, isn't it? Corona vaccine is available now. Of people who are at the age of above 45, now they are given these vaccines, right? So you must know about vaccine and what is the use of the vaccine, what is its mechanism. See what happens in the vaccine. See the slide. The principle of vaccination or immunization is based on the property of the memory of the immune system. Okay? Our immune system has memory, so it can recognize bacteria and it can memorize its structure. Okay, why it has to memorize its structure? Because if once again the same bacteria enters our body, it must be killed in the initial stage itself. That is why our immune system identifies the antigens or these bacterial cells. See, during the process of vaccination, see the slide. During the process of vaccination, inactivated or weakened pathogens or antigenic proteins of the pathogens are introduced into the body of the host. So nothing but the vaccine contain the actual virus, which is weakened, which is inactivated. Suppose the corona vaccine is there. In the corona vaccine, what do they give? They inject the vaccine only. They inject the coronavirus itself, but the virus which is there is inactivated or they are weakened. Okay, these weakened or inactivated viruses are injected as the vaccines. Okay, they initiate. Why these inactivated viruses have to be injected? Because see the slide. They initiate the production of appropriate antibodies in the host and also generate memory B cells and memory T cells. What do? They help in producing the antibodies. So these inactivated pathogens trigger the immune system. They trigger the production of the antibodies against those viruses. Okay? So within 1 week or 2 weeks, our body, the person who is immunized, he develops the antibodies, and these antibodies get ready for the attacking, for the protecting the body from that virus. On subsequent exposures, the memory cells recognize the pathogen quickly and overcome the invader with a rapid and massive production of antibodies. The people who are immunized or vaccinated, people who have taken vaccine, they have memory cells inside them. Since they have memory cells for the virus, whenever the virus enters in future, these memory cells immediately identify them and start the production of antibodies. Since they are identified very in the earlier stage, person will not be affected by any symptoms. Okay, and he will be safe. That is the use of vaccine. Okay, vaccines, what do? They trigger the immune system and they produce the antibodies in the body. Let's see the next 2 marks question. Mention various types of immunological disorders. Okay, there are disorders which are caused by immune system. Okay, what are they? First is immunodeficiency disorders. Okay, second, hypersensitivity disorders. If the reactions are continuously happening with more rapid, more than required, that's called hypersensitivity. And autoimmune disorders. Autoimmune disorders means our immune system works against us. That's called autoimmunity, autoimmune disorders. Okay? If immune cells are destroying our own cells, our own body cells, that kind of a disorder is called as autoimmune disorder. Let's go to the next question. What are autoimmune disorders? Give 2 examples. Let's know the definition of it. Generally, our immune system can recognize our own proteins and does not attack on own tissues. This is what I shown you on the slide, the first slide, isn't it? Our immune system, what it does, it identifies self and non-self substances. Self substances are saved and non-self substances are destroyed, isn't it? But unfortunately, see the slide. Unfortunately, in some cases, our immune system fails to recognize some of our own body proteins and treats them as foreign antigens. Okay, that results in attack on our own tissue. This leads to autoimmune disorders. Okay, so if our own immune system fails to recognize our own body tissues or body cells, that's called autoimmune disorder. Okay. Examples for these diseases are Graves' disease and rheumatoid arthritis. These are the two examples for the autoimmune disorders. It's most important. Many times it is repeated as well. So learn it. Next, let's go to the next question. How can the graft rejections be avoided in patients? Graft rejection means what? If any person is undergoing the organ transplantation, suppose a person requires an organ like a kidney, if any donor is donating him, so if the transferring the organ inside the patient, sometimes the organ transplantation fails. The failure of organ transplantation is called graft rejection. Okay, how it can be prevented? Let's see. Tissue matching and blood group matching are essential before undertaking any graft or transplant. Even after this, the patient has to take immunosuppressant drugs throughout their life. Okay, so blood matching, they will check the blood group matching. Okay, and tissue matching, both are checked in the two patients, the donor and the recipient, in both. Okay, and after this they take immunosuppressant drugs for the protection. Okay, this is how the graft rejections can be avoided in the patients. Go to the next question. Describe the various types of barriers of innate immunity. This is for 4 marks. Okay, so far we have learned the 2 marks questions, 8 questions we completed. Now let's see some of the 4 marks questions. Innate immunity. Innate immunity means the immunity which is there in the body by birth, from the birth. Okay, that's called innate immunity. It is not acquired. Acquired immunity means that we get after the birth. Innate immunity means the immunity we got by birth. Let's see what is that. The inborn resistance to diseases possessed by all the living organisms is called innate immunity. Different types of barriers of immunity are given below. First is physical barriers. Okay, if any foreign antigens or the bacteria, viruses attack our body, we have barriers to fight them against. Okay, so what are they? Primarily, the first barrier is the physical barrier. What are they? Our skin and mucous membranes are the main physical barriers. See the slide. Skin prevents the entry of microorganisms, whereas mucous membranes help in trapping the microbes entering our body. Next is physiological barriers. Physical barriers are skin and mucous membrane. Physiological barriers are the secretions of them. Secretions of the body like HCl, hydrochloric acid, in the stomach, saliva in the mouth, tears from the eyes are the physiological barriers against the microbes. So these HCl kills the bacteria, and saliva in the mouth, it has lysozyme enzyme. Lysozyme kills the disease causing organisms. Tears also contain this lysozyme. Okay, let's go to the other physiological barriers. Saliva, tears, sweat contain lysozyme. Just now I told, which can digest bacterial cell walls. Oil or sebum, sweat of skin, pH ranges 3 to 5, making the skin an effective inhibitor of bacterial growth. That's why, since our skin secretes mucus, our skin secretes sebum and sweat, our skin cannot develop any bacteria. Okay, but it is against to bacterial growth. Let's see the next barrier, that is cellular barriers. Polymorphonuclear leukocytes, PMN. These are neutrophils. Monocytes and natural killer cells in the blood, as well as the macrophages in the tissues, are the main cellular barriers. They phagocytose and destroy the microbes. So what do? They engulf the bacteria and they kill it. If the bacteria or the disease causing organism reaches to the blood, these cells help in the attacking them. Next, cytokine barriers. The cytokines secreted by the immune cells are involved in differentiation of the cells of the immune system and protect the non-infected cells from further infection. Okay, these are cytokine barriers. Let's see the next question. Explain the mechanism of vaccination or immunization. Okay. This is asked in the 2 marks and also asked in the 4 marks. Let's see. The principle of vaccination or immunization is based on the property of the memory of the immune system. During the process of vaccination, inactivated or weakened pathogens or antigenic proteins of the pathogen are introduced into the body of the host. They initiate the production of appropriate antibodies in the host and also generate memory B cells and memory T cells. On subsequent exposures, the memory cells recognize the pathogen quickly and overcome the invader with a rapid or massive production of antibodies. Okay, we have studied this. Let's see the next question. Explain the B cells or B lymphocytes. They develop in the bone marrow. This is the first point regarding the B lymphocytes. It is the most important question, dear students. They develop in the bone marrow. These cells are activated on their encounter with foreign agents. These foreign particles act as foreign markers. Okay? Next, the B cells immediately differentiate into plasma cells, which produce antibodies specific to the foreign particle or so-called antigen. Next, these antibodies attack to the surface of the antigen or foreign agent. Next, these antibodies detect any antigen in the body and destroy it subsequently. The immunity dependent on B cells is called humoral immunity. See, the B cells are also called as bone marrow lymphocytes. These bone marrow lymphocytes help in identifying and also producing of the antibodies. Okay, B cells produce the antibodies, remember. And B cells activation or B cells immunity is so specific. Let's see the comparison between B cells and T cells. T cells means the thymus lymphocytes, B cells means bone marrow lymphocytes. T cells originate in the bone marrow, whereas B cells originate in the both origin in the bone marrow itself, but T cells mature in the thymus. But whereas B cells mature in the bone marrow itself, and in the birds it is in the bursa fabricus. These T cells are long-lived, whereas B cells are short-lived. Okay. T cells are highly mobile. That means they move. They move from one place to another place. But B cells fairly mobile. They are stationary mostly. No complement receptors are seen in the T lymphocytes, whereas complement receptors are seen in the B cells. There is no surface immunoglobulins in the T cells, but surface immunoglobulins are existing in the B cells. Why? Because B cells help in the specific immunity. Okay, they produce antibodies. There is no antibody synthesis in the T cells. Thymus lymphocytes do not produce any antibodies, but whereas B cells produce antibodies. Okay? Antibodies are specific. Next, effector, where they affect cellular and humoral effect. And here in the B cells we see only humoral effect. Okay. Antibodies. Write short note on immunoglobulins. This is also again a most important 4 marks question. See this, I'll explain it as well. See the slide. They identify the large target pathogen by binding to the antigen. See here in the picture, you can see antibodies are getting binded to the antigen, isn't it? Antibodies are specific to its antigen. For example, antibodies produced against one bacterium cannot act on against the other bacterium. So they are specific why antibodies produced against one bacterium cannot act against the other bacterium. The two types of antibodies are free or circulating antibodies and surface antibodies. Surface antibodies are located on B cells, memory cell surface, which are useful for identification purpose. Okay. Antibodies are the army of proteins produced by lymphocytes to fight against the pathogens. Let's see immunoglobulins. Antibodies are also called immunoglobulins. This is the structure of them. Structure of antibodies proposed by Ron D. Porter. Ron D. Porter is proposed structure. It's a Y-shaped structure made of 4 polypeptide chains, which two are long. In the 4 polypeptide chain, 2 chains are long. Here in the picture you can see, this is the one chain, this is another chain. The chain which is longer is called a heavy chain, and chain which is short is called as light chain. So the upper region is called variable region and the lower region is called constant region. Okay. There are 2 heavy and 2 light chains, hence represented as H2L2. These are bound by disulfide bonds. Here you can see they are bound by disulfide bonds. The structure is called H2L2. H means heavy chains, L means light chains. Okay. Antibody molecules has 2 ends, FAB end, antibody end, that's called fragment antibody binding, and FC end, fragment crystallizable fragment cell binding end. Okay. So here in the picture you can see the antigen and antibody. So paratope is the antigen binding site of the immunoglobulin is called as paratope. Okay. Epitope means the surface of the antigen which is binding to the paratope. So epitope binds to the paratope. Okay. Hope you understood epitope and paratope as well. Let's go to the next question. Explain humoral immunity. So there are 2 kinds of immunity in the process of immune mechanism. Okay. So now we will see what is meant by humoral immunity. There are certain steps for humoral immunity. In secondary lymphoid organs, the free antigens of pathogens bind to antigen presenting cells. This is the first step in which antigen presenting cells identify the antigens. Next, B cells engulf and process the antigens and present with the help of MHC class 2 molecules. I will show you the pictures also regarding this humoral immunity. Then you will understand what is the actual process of it. Okay. T helper cells interact with MHC class 2 molecules and release interleukins. So T helper cells interact with MHC class 2 molecules. These MHC class 2 molecules are existing on the surface of the cells. Okay. MHC means major histocompatibility complex. Interleukins stimulate the B cells to differentiate into memory cells and plasma cells. Okay. Interleukin cells help in stimulation of B cells. The plasma cells release specific antibodies into the plasma or extracellular fluid. Antibodies kill the bacterium by neutralization, agglutination, and precipitation, which leads to the opsonization and phagocytosis. Let's see the process now. See in the step 1 what is happening. So bacteria is identified the APC cells, that is antigen presenting cells. Okay. After the breaking down of the bacteria, what is happening? It is processing. Processing means the killing of bacterium. Okay. Next is presentation. In the presentation, The cell surface has MHC class 2 molecules, which are major histocompatibility. With the help of MHC class 2 and presenting antigens, they present the antigen determinants to T helper cells. Here you can see in the first phase, what is happening? Phagocytosis. Bacteria is getting inside and the bacteria is broken. The breaking of the bacteria is called as processing. First step is phagocytosis. Second is processing. In the same cell, the processing take place. After the processing, whatever the determinants are there, whatever the broken substances are there, they are presented by the MHC class 2. Okay, these MHC class 2 present these antigen determinants, and T helper cells identify these bacteria by binding to the MHC class 2. Okay, so see here, T helper cells are identified it. See next what happens in step 2. Step 2 and 3. See, when the antigen determinants are identified by the T helper cells, they interact with the MH class 2 cells, isn't it? So soon after identification of the antigen determinants, these T helper cells start secreting the interleukins. Okay, so these interleukins help in signaling to the B cells. So the first step is phagocytosis, second is presentation, third is the second is the processing, third is the presentation, fourth is the interaction with the T cells, and fifth step is releasing of interleukins. Let's see in the fourth and fifth step what happened. These interleukins which are released by the T helper cells, they stimulate the B cells differentiation. When B cells are differentiated, two cells are produced. One kind of cell is a plasma cell and second is memory cell. Plasma cell of the B lymphocytes start producing the antibodies. This is how the process goes on. Okay, so hope you understood. In the last, in the first step, you can see phagocytosis. Second step is these bacteria are killed. This is called processing, and the broken determinants of the bacteria are presented. This is the third step. And fourth, these broken cells are identified by the T helper cell. That is the third step. And these after identification, they release the interleukins. Interleukins stimulated the B lymphocytes, and B lymphocytes started differentiating into two cells, plasma cell and memory cells. And finally, from plasma cell, the antibodies are produced. This is how the process takes place. The whole process is called as humoral immunity. Okay, hope you understood the humoral immunity. Let's go to the next question. Cell mediated immunity. Says there is no antibodies involved here, only cells act on this immunity. Okay, cells produce certain substances, and these substances help in killing the foreign particle. The immunity, this immunity is mediated by activated T lymphocytes and natural killer cells. It does not involve in the production of antibodies. Okay. So in the humoral immunity, there is a production of antibodies, and in the cell mediated immunity, there is no production of antibodies. It is effective against both exogenous and endogenous antigens. Let's see the steps. First is antigen presentation. Antigen presenting cells phagocytose the pathogens and present antigen fragments with the help of MHC class 2 molecules. Second, release of cytokines. Okay, instead of interleukins, here the cytokines are secreted. What do these cytokines do? T cells bind to MHC class 2 molecules and produce interleukins. Okay, so interleukins are a form of cytokines. Next, see, activation of cytotoxic T cells. Interleukins stimulate the T cytotoxic cells to get activated, and the T cells binding with infected cell. After binding to infected cells, what happened? Activated cytotoxic T cells binds to the infected cells and release perforins and granzymes. Perforins and granzymes, let's see what they do. After the release of these perforins and granzymes, the destruction of the infected cell takes place, which is called as apoptosis due to perforins and granzymes. What do? Perforins, they form the holes to the infected cell. Perforin means the hole. Okay. Next, granzymes enter the body and destroy it. Okay. So perforins helpful for making the holes to the infected cell, and granzymes they enter inside and destroy complete cell. See here, the cell-mediated immunity. First is antigen presentation. When the pathogen comes, the phagocyte recognizes it and presents to the T helper cell. T helper cell is identified the antigen determinants or the pathogen determinants, and they start secreting the cytokines, which are the interleukins. These interleukins, what do? They help in activation of cytotoxic T cells. Cytotoxic T cells, they bind with the infected cell with the help of MHC class 1 molecules. Then they starts producing perforins and granzymes. Perforins, what do? They make the holes to the cell, and granzymes enter inside and destroy it. This is how the cell death take place. The cell death is called as apoptosis. Okay. Hope you understood the whole process of cell-mediated immunity. Cell-mediated immunity is different from humoral immunity because in the humoral immunity there is production of antibodies, but in the cell-mediated immunity there is no production of antibodies, but there is a production of granzymes and perforins. Okay, in humoral immunity we have seen that MHC class 2 molecules are only used. Okay, but in cell-mediated immunity, MHC class 1 molecules are also used. These MHC class 1 and class 2 molecules are existing on the surface of the cells. Okay. See differences between humoral and cell-mediated immunity. See here, main cells involved in the humoral immunity, B cells, and in the cell-mediated immunity, T cells. Where do the cells develop? In the humoral immunity, it is produced and matured in the bone marrow, whereas in the cell-mediated, produced in the bone marrow and mature in the thymus. Antibodies involve production of antibodies and does not involve the production of antibodies. How are pathogens are identified? In the humoral immunity, we have seen that via antigens floating in the blood. Okay, antigens are nothing but the pathogens, which are disease-causing microorganisms. Next, in the cell-mediated immunity, antigens are on the cell surface of the infected cells. How are pathogens killed? Here in the humoral immunity, since there is a production of antibodies bind to the the pathogen. Pathogen means disease-causing organism and kills it. Okay, but here in the cell-mediated immunity, it kills the infected cell by killer T cell. Okay, how do cells divide once they are stimulated? Cells divide either plasma cell or the memory cell. Okay, but in cell-mediated immunity, cells divide into different types of specialized T cells, and then they start producing perforins and granzymes. These are the some differences between humoral and cell-mediated immunity. Both are not the same. Okay, you should know this. Next, let's practice some neat questions. Okay, so previously from this chapter, some of the questions are asked. Okay, we will see what are those questions. If by watching those questions, by learning those questions, you can estimate the way of asking questions. Okay, so that you will prepare in that manner. Okay. Let's see the first question. If you suspect major deficiencies of antibodies in a person, to which of the following would you look for confirmatory evidence? Okay. It is asked in the NEET 2015. Option A, serum globulins. B, fibrinogen in plasma. Serum albumin and hemocytes. What is the answer? Can you guess the answer? Yes, the answer is serum globulins. Okay, this is the right answer regarding first question. Let's go to the next question. Which of the following immunoglobulins does constitute the largest percentage in human milk? Just now we have learned in this class itself. Can you guess the answer? Option A, immunoglobulin M. Option B, IgA. Option C, IgD. Option D, IgE. The answer is immunoglobulin A. Okay, remember. Let's see the next question, which is asked in the NEET 2016. Anti-venom injections contain preformed antibodies, while polio drops that are administered into the body contain. Okay, so we must know what polio drops comes under. Are they attenuated pathogens, activated pathogens, gamma globulins, or harvested antibodies? Can you guess the answer? Yes, the answer is attenuated pathogens. Attenuated means weakened. They are weakened or inactivated pathogens. Okay. Let's go to the next question. In higher vertebrates, the immune system can distinguish between its own cells and foreign cells. If this property is lost due to genetic abnormalities and it attacks its own cells, then it leads to, okay, if it goes wrong and attacks own cell, what happens? See the options: activated immunity, graft rejection, autoimmune disease, none of the above. Can you guess the answer? We have studied just now. Yes, the answer is autoimmune disorders or autoimmune disease. Autoimmune means ability which is, sorry, it is abnormality of identifying the self substances of the immune system. Okay. Asthma may be attributed to options: inflammation of the trachea, accumulation of fluid in lungs, bacterial infection of lungs, allergic reaction of the mast cells in the lungs. Can you guess the answer? Yes, the answer is allergic reaction of the mast cells in the lungs. Due to these allergic reactions, asthma take place. Okay, these allergic reactions may take place with the help by the environmental pollution and also by pathogens. Let's go to the next question. Which of the following sets of diseases are caused by bacteria? Tetanus and mumps? No, this is not. Herpes and influenza? No, influenza caused by virus. Typhoid and smallpox, cholera and tetanus. Which one is the answer? The answer is cholera and tetanus. Okay. Which of the following disease is caused by a protozoan? Okay, which is caused by protozoan? Right? Influenza, cancer, syphilis, and babesiosis. Babesiosis. Okay. The answer is babesiosis. Right? This is caused by a protozoan. It is asked in the NEET 2015. Let's go to the next question. Grafted kidneys may be rejected in a patient due to why the graft rejections take place. See the options: cell-mediated immune responses, passive immune responses, innate immune responses, or humoral immune responses. Can you guess the answer? Yes, the answer is cell-mediated immune response. See, the cell-mediated immune response is responsible for the graft rejections. Okay. Let's go to the next question, which is the 9th one. The active form of Entamoeba histolytica feed upon. So this Entamoeba histolytica, you have studied about this particular pathogen, which is from protozoa in the first year, isn't it? So where do they, what do they feed upon? They only blood. Option B, erythrocytes, mucosa and submucosa of colon, mucosa and submucosa of colon only, or feed in intestine? What do you think? Which one is the answer? Yes, the answer is erythrocytes, mucosa, and submucosa. Okay. So the Entamoeba histolytica feeds upon RBC, mucosa, and submucosa of the colon. Mucosa and submucosa are the internal layers of the large intestine. Okay. Colon is a part of the large intestine. Okay. Let's go to the last and the 10th question. The cell-mediated immunity inside the human body is carried out by, what do you think? Just now we have studied about both cell-mediated and humoral immunity. The options are thrombocytes, erythrocytes, T lymphocytes, and B lymphocytes. What do you think? The answer is T lymphocytes, exactly. Because B lymphocytes are involved in the humoral immunity, T lymphocytes are involved in the cell-mediated immunity. Okay, just now we have studied this. Okay, dear students, thank you for watching this class. Hope you understood the class completely. I wish you all the best for your future preparation. Thank you, dear student. Thank you.
The number you have dialed is currently busy. Please try later. మీరు డైల్ చేస్తున్న నంబర్ ప్రస్తుతం బిజీగా ఉంది. రైట్